Welcome back to the Deep Dive, where we take the immensely complicated systems that govern our world, or in this case, govern you, and distill them down into essential, fascinating knowledge. Today, we are undertaking a deep dive into the very architecture of consciousness and action. We're talking about the nervous system.
This is truly the ultimate deep dive. We're talking about the body's electrical grid, the system that allows for, instantaneous communication, computation, and control. There's a great quote from Peter D. Kramer that really frames the challenge. He said, "If the human brain were simple enough for us to understand, we would be too simple to understand it."
That perfectly captures the paradox, doesn't it? And our mission today is to try and understand the language of that system. We're going to look at how electrical signals are generated, how they're transmitted, and how they're interpreted. And we're really starting from square one today, assuming you, our listener, are completely new to this. We're building the signal from scratch, step by step.
That's the plan. And to start, we have to appreciate the job description. The nervous system exists to perform three core continuous functions. It's operational trilogy: sensation, integration, and response.
Okay, let's unpack that trilogy. If we think about the nervous system as like the ultimate information processor, what's the input stage?
The input stage is sensation. It's the process of receiving information about the environment. And this input can come from outside the body: what you see, what you hear, taste, touch, smell.
Classic five senses.
Right. But critically, it can also come from inside the body. Your nervous system is constantly monitoring your internal world. Things like the stretch of your organ walls, your blood pressure, or the concentration of key ions like sodium in your blood. Any measurable change, internal or external, that's what we call a stimulus.
So our five major senses are just the tip of the iceberg of this constant stream of sensory input. The system is always running diagnostics in the background.
Yeah.
Once that raw data, that stimulus is registered, what happens next?
That data gets sent to central command. That's the brain and the spinal cord, which together we call the central nervous system, or CNS. And the next step is integration.
The processing phase.
Exactly.
Yeah.
This is where the system compares the new sensory input with everything it already knows. Stored memories, your current emotional state, and anything you've previously learned, which we just group under the term cognition.
So integration is the CPU, the critical thinking phase. It's not just recognizing data. It's putting it into context and deciding what to do about it.
Perfect. Let's use that classic baseball analogy. The sight of a fastball hurtling toward the plate, that is sensation. But a veteran batter, they don't just automatically swing. Their brain is instantly integrating. It's asking, what is the score? That's cognition. Is this a high-stress moment? That's your emotional state.
Right.
Have I seen this pitcher use this pitch successfully before? That's memory. This complex, simultaneous calculation, it takes place in what we call the association areas of the cortex. And the output of that calculation is a complex decision, which then dictates the response.
Which brings us to the final piece of the trilogy, response. The system has sensed, it's integrated, now it has to execute that decision.
The response is the motor output. It's the action generated based on that integration. And the system is a master controller of the body's effectors. It can cause the contraction of skeletal muscle, like swinging the bat, or cardiac muscle, controlling your heart rate, and smooth muscle, controlling blood flow or digestion. It also controls glandular output, like activating sweat glands to regulate temperature.
And when we look at those responses, we can categorize them based on how much conscious control we actually have over them.
Precisely.
Mhm.
We divide the nervous system functionally based on the nature of that motor response. So first, we have the somatic nervous system, or SNS. This is the system associated with conscious perception and voluntary motor responses. This is primarily the movement of skeletal muscle when you deliberately decide to walk or talk, or in our baseball analogy, to swing.
That's the system of deliberate action.
Mostly. It also handles some reflexes, like the knee-jerk response, which sort of bypasses the conscious integration step, but still uses skeletal muscle to act.
Okay, so that's the voluntary system. Then there must be an involuntary one running in the background.
And there is. That's the system running beneath the surface, maintaining internal balance. The autonomic nervous system, the ANS, this is all about involuntary control. Its main job is regulating the internal environment, maintaining what we call homeostasis. It controls your cardiac muscle, your smooth muscle, and your glandular tissue. For example, it automatically increases your heart rate and sweat production when you start exercising. You don't have to consciously command your heart to speed up. The ANS just handles it.
And we often hear about a special sort of third autonomous network that's dedicated just to the gut.
That's the enteric nervous system, the ENS. It's a specialized network of neural tissue that's woven throughout the walls of the digestive tract. It controls smooth muscle contraction and gland secretion completely on its own. It's often categorized as a subdivision of the ANS, but it operates with remarkable independence.
So from sensing hot coffee to deciding whether to put it down or drink it slowly, it all flows through sensation, integration, and response, orchestrated by these different systems. But how is that information, the decision to drink the coffee or drop it, how is it physically transmitted? That brings us to the actual components. Nervous tissue. Okay, so we know the nervous system's job. Now let's look at the actual building blocks, the hardware responsible for carrying and processing those electrical messages. What are the key players in nervous tissue?
There are two fundamental cell types. First, and the most famous, are the neurons. These are the functional workhorses. They're specialized for electrical signaling, for computation, and for rapid communication across long distances. They are electrically active cells that form these complex networks and communicate by releasing chemical signals.
And the second group, the unsung heroes.
Those would be the glial cells, or just glia. The name comes from the Greek word for glue, which is a pretty good analogy. They were initially thought only to hold things together, but we now know they are essential support cells. They provide the structural framework, they maintain the precise chemical environment that neurons need to function, and they generally just facilitate the neuron's job. Without glia, neurons fail very, very quickly.
The sources outline a few specific types of glia that have really specialized roles, particularly when it comes to insulation and protection. Let's start with protection inside the brain and spinal cord.
Right, in the CNS. A critical protective cell here is the astrocyte, so named because of its star-like shape. Astrocytes provide metabolic support, regulating the concentration of ions and neurotransmitters in the extracellular space. But their most famous job is contributing to the blood-brain barrier, or BBB.
The BBB. That sounds crucial. Is it like a physical wall?
It's more complex than a wall. It's a physiological restriction. Capillaries in the brain are wrapped very tightly by the end feet of these astrocytes. And this greatly restricts which substances circulating in the blood, including many drugs or pathogens, can actually pass into the delicate neural tissue. The astrocyte is effectively the gatekeeper, or maybe the bouncer, ensuring only very specific nutrients get through while keeping toxins out.
That makes perfect sense. The brain is far too vital to risk contamination. Now, what about the glial cells that handle the insulation?
Right, we need insulation to speed up those electrical signals. That insulating substance is called myelin. In the CNS, the myelin is provided by the oligodendrocyte, a beautiful name that means cell of a few branches. And the fascinating detail here is that just one oligodendrocyte can extend its processes to wrap and insulate segments of multiple different axons. It's a multitasker.
And what about the system that's running outside the brain and spinal cord, the peripheral nervous system, or PNS? Are the support cells different out there?
They're structurally different, which reflects the different environment. In the PNS, the myelin is provided by the Schwann cell. And the key distinction is that a single Schwann cell typically wraps around a portion of only one axon segment, much more focused.
Okay, so the neurons are the messengers, and the glia are the complex support staff. Let's zoom in now on the neuron itself and see how it's designed to handle this electrical flow, which is all dictated by its structure, its polarity.
The basic structure dictates the direction of information flow. At the center is the soma, or the cell body. This contains the nucleus, mitochondria, all the standard organelles. This is the neuron's factory and its command center.
And where does the information arrive?
It arrives at the highly branched processes that extend from the soma, which we call dendrites. Think of dendrites as the neuron's highly sensitive receiving antenna. They are specialized to receive input signals, usually from other neurons, at these junction points called synapses. A single neuron can receive thousands of inputs across its dendrites.
That information then flows through the cell body. Where does it decide whether to fire or not?
That decision point is the single most critical location on the entire neuron. The initial segment, which is also famously known as the axon hillock, this is the tapering region where the single long process, the axon, emerges from the cell body. If the neuron decides to fire, the electrical signal, the action potential, is initiated right here and then travels down the axon.
So the axon is the transmission line, carrying that electrical signal away from the soma toward target cells. The flow is strictly one way. Dendrite to soma to axon hillock to axon. That polarity is fundamental.
It is the immutable law of the neuron. And now we shift to the core mechanism. How do neurons maintain and use an electrical charge? They have what we call an excitable membrane.
What makes a neuronal membrane excitable?
The excitable membrane is essentially the lipid bilayer of the neuron, but it's studded with these specialized proteins designed to rapidly and dynamically regulate the movement of charged particles, or ions. The distribution of this charge across the membrane is the membrane potential, and we measure it in millivolts, or MV. And by convention, we always measure the charge inside the cell relative to the outside.
And in the quiet, stable state, when the cell isn't firing, what is that charge?
That is the resting membrane potential, and it is typically maintained at negative 70 millivolts. Negative 70 millivolts. That means the inside of the cell is significantly more negative than the outside.
Okay, why that specific number, negative 70, and how is it maintained? It seems like it would take a lot of work.
It's a remarkable balancing act maintained by two key factors, and yes, both involve significant energy expenditure. The first is the powerhouse protein called the sodium-potassium pump. This is an ATPase, which means it uses ATP, the cell's energy currency, to actively move ions against their concentration gradients.
And what's the exchange rate for this pump?
The pump actively moves 3 positively charged sodium ions, Nawu, out of the cell for every 2 positively charged potassium ions it moves in. Think about that ratio for a second. 3 positive charges out, only 2 positive charges in. It is constantly ejecting more positive charge than it brings in. This creates a net electrical imbalance, making the inside negative, and at the same time it maintains a steep chemical gradient. Sodium is kept high outside and potassium is kept high inside.
So the sodium-potassium pump is constantly charging the battery, and it's very expensive to run. What's the second factor that makes this necessary?
The second factor is the presence of leakage channels. Even when the neuron is at rest, ions are slowly leaking across the membrane through these channels that just open and close randomly. Specifically, the cell membrane is much leakier to potassium, to Co, than it is to NO. Since Co is high inside, Co is constantly slowly leaking out. And because it's a positive charge leaking out, this constantly drives the membrane potential a little bit more negative. The sodium-potassium pump has to continuously work just to correct this relentless leakage and keep the voltage stable at negative 70.
That makes the pump sound less like the creator of the charge and more like the constant maintenance crew needed just to stay at negative 70 milliVed.
That's a perfect way to put it. Now, to use that stored electrical energy, the ions have to move quickly across the membrane. But because they're charged, they can't penetrate the greasy lipid core of the cell membrane. They need assistance from channel proteins, which act as specific doors.
And those doors are incredibly sophisticated. They don't just let anything pass.
Absolutely not. Channel proteins enforce 2 strict rules of entry. First is electrochemical exclusion. The channel is selective based on the ion's charge. For example, a channel that lets positive ions, or cations, through will have negatively charged amino acids lining its pore to attract them, and vice versa.
And the second rule?
Size exclusion. The channel's diameter is precise, calibrated to the ion's hydrated diameter, that's the size of the ion, plus the water molecules clinging to it. If the ion is too big, or if it sheds its water molecules, the pore won't let it through.
So if the ion is not the right size, or the right charge, the door stays shut. But to create a signal, we need doors that are regulated, the gated channels.
Right. And we classify these gates by what event opens them. The first type is the mechanically gated channel. These open due to physical distortion of the cell membrane, things like stretching, pressure, vibration, or temperature changes. When you tap your finger, the pressure deforms the sensory receptor ending, and that opens these channels.
A physical stimulus opening an electrical door. What about a chemical stimulus?
That leads to the ligand-gated channel, which is also known as an inotropic receptor. A ligand chemical signaling molecule, often a neurotransmitter in the nervous system, binds to the channel protein like a key fitting a specific lock. This binding causes an immediate conformational change, and the channel opens instantly, allowing ions to cross.
And the final type, which defines the electrical signal, we call the action potential.
That's the voltage-gated channel. These are found clustered heavily at the axon hillock and all along the axon. They are regulated entirely by the electrical status of the membrane. When the voltage inside the cell changes, specifically when it becomes less negative than negative 70 milliV, these channels sense the shift and are prompted to open. These voltage-gated channels are the true drivers of long-distance communication.
We've established the stage, the neuron resting at a negative 70 milliV charge, waiting for input via its dendrites. Now we apply stimulus. That first small change in voltage is called a graded potential.
Right, a graded potential, or GP. It's a local, transient change in the membrane potential. And the most important characteristic here is that the electrical change is directly proportional to the strength of the stimulus. It's not an all-or-nothing event yet.
So if the stimulus is weak, the graded potential is small, and if the stimulus is strong, the graded potential is large.
Exactly. Imagine a pressure receptor in your skin. A light touch opens only a few mechanically gated channels, causing a small, local voltage shift. A firm press opens many more, causing a much larger voltage shift. These GPs usually occur at the dendrites, or the sensory receptor endings, where the input is first received.
The word graded suggests the size varies, but these potentials also have an expiration date, right? They don't last forever.
They do not. Graded potentials are local. They only spread a very short distance before they fade out, and they're transient, they disappear quickly. If they're going to trigger a major signal, they need to happen near the axon hillock, and they need to add up fast.
And they can push the neuron in two opposite directions. They can excite or they can inhibit.
Yes. If the membrane voltage moves from -70 milliV towards zero, becoming less negative, say, to -65 milliV, that is a depolarizing graded potential. This typically happens when positive ions, like sodium or calcium, rush into the cell. When this specific depolarization occurs at a synapse, it is called an excitatory postsynaptic potential, or EPSP. It moves the neuron closer to its firing threshold.
The system is revving its engine, making an action potential more likely. What about applying the brakes?
That's the hyperpolarizing graded potential. In this case, the voltage moves below the resting state, becoming even more negative, for example, to -75 milliV. This usually happens when positive ions like potassium rush out of the cell, or when negative ions like chloride rush in. If this occurs at a synapse, it's an inhibitory postsynaptic potential, or IPSP. It actively suppresses the neuron, pushing it further away from the firing threshold.
This sounds like the neuron is constantly receiving contradictory advice from thousands of inputs, some excitatory, some inhibitory. How does it make sense of all that conflicting information?
This is where the magic of the nervous system begins, and it all happens at the axon hillock. Summation. Because graded potentials are small, local, and transient, a single one is almost never strong enough to trigger the main signal. They must summate, they have to add together mathematically, to push the cell to its critical firing voltage.
And what is that critical voltage? How much change is required to flip the switch?
For a typical neuron, the total net depolarization required is generally plus 15 milliV. So if the neuron starts at -70 milliV, it has to reach -55 milliV. That -55 milliV mark is called a threshold. If the combined EPSPs and IPSPs hit -55 milliV, the cell fires an action potential. If they sum to -56 milliV, nothing happens. It is a critical line in the sand.
Okay, so if I have one EPSP that causes a +5 milliV shift and an IPSP that causes a -3 milliV shift, the net result is only +2 milliV. That
Right.
won't do it.
Precisely. You need multiple +5 milliV hits to overcome any inhibition and cross that line. And there are two main strategies for hitting that -55 milliV target. The first is spatial summation.
Spatial meaning location, so multiple places firing at the same time.
Correct. Imagine the axon hillock is the target. Spatial summation occurs when multiple graded potentials arrive from different locations, say 4 different dendrites, each receiving an excitatory input at roughly the same time. These separate small inputs converge, and their voltages are combined instantly.
Like 4 small streams combining right before they spill into a reservoir, hitting the critical level. What about the second strategy?
That's temporal summation. Temporal refers to time. This happens when multiple graded potentials arrive from the same location, the same synapse, but they arrive in quick, rapid succession. Because the first graded potential hasn't had time to fade away, the second one stacks on top of it, increasing the magnitude of the depolarization.
So the neuron is constantly solving this differential equation at the axon hillock, adding and subtracting the EPSPs and IPSPs until it either hits -55 milliV or the signals fade. It's the ultimate internal democracy.
And that calculation is the defining moment. Once -55 milliV is reached, the neuron commits.
We've established the foundation, and calculus of summation has confirmed that we have hit the magic number, -55 milliV. Now the main event fires, the digital message, the action potential.
This is the critical electrical impulse used for long-distance communication. It is, as we noted, an all-or-none event. The neuron either fires fully or it does not fire at all. There is no middle ground, and there is no such thing as a half action potential.
I think this is a point of confusion for many people. If a stimulus is incredibly strong, does the action potential get bigger? Does it hit, say, plus 50 milliV instead of plus 30 milliV?
Absolutely not. Once the threshold of negative 55 milliV is reached, the resulting action potential is always the exact same size, peaking consistently around plus 30 milliV. The strength of the stimulus is encoded not in the size of the signal, but in the frequency of the firing. A stronger stimulus will initiate multiple action potentials in quick succession, sending the message more urgently, but each individual AP will be identical in magnitude.
Okay, let's walk through the spectacular phases of this, 2 millisecond event, focusing on the movement of ions through those voltage-gated channels. We start at the resting state at nadish 70 milliV.
Correct. At rest, the critical players, the voltage-gated sodium channels, they have two gates. You can think of them as a turnstile and a blocker gate. The activation gate, which is the turnstile, is closed, but the inactivation gate, the blocker, is open. The channel is locked, but it's ready.
Then we hit the threshold at nadish 55 milliV.
And that initiates the first and fastest step, depolarization, the rising phase. When the voltage hits nadish of 55 milliV, the voltage-gated NAO activation gates snap open instantly. Because NAO concentration is overwhelmingly high outside the cell, and the inside of the cell is highly negative, NAO rushes in with incredible force. It's driven by both its concentration gradient and the electrical gradient. This massive rapid influx of positive charge causes the voltage to skyrocket from nadish 55 milliV past zero, peaking at plus 30 milliV.
That influx defines the signal spike. What stops that positive surge from just continuing?
That leads directly to the next phase, repolarization, the falling phase. This phase is triggered automatically when the voltage hits the peak of plus 30 milliV. At this exact peak, two things happen to reverse the charge. First, the voltage-gated Na inactivation gate, the blocker, quickly swings shut. This physically blocks the NAU pore, stopping the influx of positive charge entirely.
The NIO channels are now effectively closed and locked.
Yes. Simultaneously, the second critical event happens, the voltage-gated potassium channels, which were actually triggered to open way back when the membrane reached negative 55 milliV. They finally opened fully. They were dramatically slower than the NIU channels. Since Ca concentration is high inside, Ca rushes out, taking positive charge with it. This outflow of Ca rapidly drives the membrane voltage back down toward the negative resting potential.
And because those Ca channels are slow, we get that slight overshoot, the final step.
That's hyperpolarization. Those voltage-gated co-channels are slow to close, so they remain open for a brief period even after the membrane potential has passed the resting value of nag 70 milliV. This allows CO to continue exiting, causing the membrane to become briefly more negative, reaching potentials like negative 80 milliV. This overshoot period is crucial because it leads into the refractory period. Once the CO channels finally manage to close, the constant action of the NAC-CO pump quickly restores the precise ion balance, bringing the cell back to its stable negative 70 milliV resting state.
This whole sequence is done in under 2 milliseconds, and it brings up one of the most critical safeguards of the system, the refractory period. Why is it impossible, or at least very difficult, to fire a second action potential right after the first one?
The refractory period is a short interval when initiating a second AP is inhibited. Its primary purpose is to ensure the signal is unidirectional, forcing it to move only forward down the axon. We divide this period based on the state of the sodium channel's inactivation gate.
Let's start with the one where nothing can possibly happen.
That is the absolute refractory period. This spans the period from the moment the AP initiates through the majority of the repolarization phase. During this time, the voltage-gated NAHO inactivation gate is closed. Since sodium cannot enter the cell under any circumstances, no amount of stimulus, no matter how strong, can generate a new action potential. It is physically impossible.
So this absolute refractory period is the fail-safe that guarantees the AP can only move forward because the segment of the axon that just fired is temporarily unresponsive.
Precisely. Then we transition into the relative refractory period. This occurs during the tail end of repolarization and the hyperpolarization overshoot. During this time, the NAHO inactivation gates have reset, they've reopened. But the cell is either slightly below rest hypopolarized or Ca is still exiting.
What does that mean for firing a new signal?
A new action potential can be generated here, but it requires a stimulus that is significantly stronger than the one that initiated the first AP. You have to work much harder to drive the voltage up from, say, negative 80 milliV back to the threshold of negative 55 milliV. This state ensures that only an intense, urgent signal can overcome the temporary hyperpolarization and trigger a new firing sequence.
The neuron has fired the digital message. Now, that action potential must be moved rapidly down the length of the axon to the next cell. This movement is called propagation. We mentioned the refractory period, forcing that signal to move only in one direction away from the soma, but the speed of that propagation is what separates a lightning-fast reflex from a slow, painful reaction.
Speed is everything, and the velocity of propagation depends primarily on two factors, the presence of myelin and the axon diameter.
Let's revisit myelin. It's that lipid-rich insulating substance. How does insulation specifically accelerate the signal?
Myelin acts exactly like the plastic or rubber coating around a copper wire. Lipids are electrical insulators, so the myelin sheath wrapped around the axon prevents the electrical current from leaking out of the cell membrane. This forces the electrical charge to spread much more efficiently and quickly down the length of the axon.
And as a reminder, we have the oligodendrocyte providing this coating in the CNS and the Schwann cell in the PNS. How does the presence of that coating change the way the AP travels?
We have two modes of conduction. First, continuous conduction, which happens along unmyelinated axons. It's slow because the action potential has to be regenerated at every single tiny segment of the axon. Voltage-gated NIU channels must open sequentially all along the entire length. Imagine trying to start a domino cascade where you have to reset each domino before the next one can fall. It's energy-intensive and slow.
The opposite is the fast leaping signal.
That's saltatory conduction. The word saltare literally means to leap or to dance. This happens along myelinated axons. The myelin covers most of the fiber, but it leaves critical tiny gaps called the nodes of Ranvier. And the crucial detail is that all the voltage-gated NAL and Chi channels are densely clustered only at these nodes.
So the signal doesn't have to waste time opening gates along the insulated segments.
Correct. The current travels almost instantaneously under the insulated sheath to the next node. The action potential is only regenerated at the nodes of Ranvier where the channels are. It effectively jumps from node to node. This mode of conduction is orders of magnitude faster and significantly more energy efficient than continuous conduction, as the Na-Ka pump only has to work at the nodes to restore ion balance.
So myelin saves time and energy, but you also mentioned the physical size of the highway matters.
The second factor is the axon diameter. This is a principle of electrical physics. Resistance. Just like water flows faster through a wide pipe or river than a narrow one, electrical charge diffuses faster inside a thicker axon because ions encounter less internal resistance. Axons can range from tiny 1 micrometer diameters up to 20 micrometers. Therefore, the absolute fastest signals, like those controlling your skeletal muscles, travel down axons that are both thick and heavily myelinated.
When this beautiful system of insulation and speed breaks down, the consequences are immediate and severe. Let's delve into the clinical connection. Demyelinating diseases.
Demyelinating diseases are the physical manifestation of signal speed failure. They occur when the immune system or other factors destroy that crucial myelin sheath, compromising electrical signaling. We see two classic examples that highlight the distinction between the CNS and PNS glial cells.
Let's start with the one that affects the central nervous system.
That is multiple sclerosis, or MS. MS is an autoimmune disorder where the body's immune cells mistakenly attack and destroy the myelin produced by the oligodendrocytes in the CNS. The damage leaves scar tissue, which is what sclerosis means, on the white matter of the brain and spinal cord.
And since the CNS handles integration and central command, what are the resulting symptoms?
The symptoms are highly varied, reflecting where the demyelination occurs, but they are often debilitating. You see issues with motor control weakness, spasticity sensory deficits like numbness or pain, and critical autonomic dysfunction like vision problems and fatigue. The signal transmission literally slows down so much that coordinating thought and movement becomes agonizingly difficult.
And the version that affects the peripheral system?
That's Guillain-Barré syndrome, or GBS. This is a rapid onset, usually temporary autoimmune disorder, where the immune system attacks the myelin provided by the Schwann cells in the peripheral nervous system.
How does that clinical picture differ from MS?
GBS typically presents as ascending paralysis, meaning weakness and tingling sensations often begin in the feet and legs and spread upward, sometimes rapidly involving the respiratory muscles. Since the PNS carries the sensory input and the final motor command out to the muscles, the breakdown of Schwann cell myelin compromises both sensation and voluntary movement, often leading to acute motor failure. These conditions really underline that the electrical language is only as reliable as its physical infrastructure.
The action potential has traveled at lightning speed down the myelinated axon and has reached the very end, the axon terminal. The message is electric, but the gap between neurons is filled with fluid. How does the electrical message jump that gap?
It doesn't jump. It has to be converted. This is the job of the chemical synapse, which is the standard junction point in the nervous system. We convert the electrical signal into a chemical signal, a neurotransmitter, which bridges the small gap, the synaptic cleft, and then converts back into an electrical signal in the receiving cell.
This sounds complex. Let's follow the flow step by step, focusing on what happens at the presynaptic element, the axon terminal.
Step 1. The propagating action potential. That depolarization wave reaches the end of the terminal. Step 2. That change in voltage opens specialized voltage-gated calcium channels in the terminal membrane.
Wait, why is calcium the key ion here? Ca, not sodium or potassium.
That's a great question. Calcium concentration is meticulously maintained at an extremely low level inside the cell, but it's high outside. When the voltage-gated Coheney channels open, calcium ions rush in down a massive electrochemical gradient. This rapid influx of calcium is the definitive trigger. It's the crucial link between the electrical signal and the chemical release.
So the calcium is the signal that says, release the message. How does it do that?
Step 3. The calcium ions bind to specific sensor proteins associated with the neurotransmitter vesicles. These vesicles are tiny membrane sacs that package thousands of neurotransmitter molecules. Step 4. Cala binding facilitates the fusion of the vesicle membrane with the presynaptic membrane through a process called exocytosis. This fusion instantly dumps the neurotransmitter molecules into the synaptic cleft.
Now the chemical signal is bridging the gap.
Step 5. The neurotransmitter molecules diffuse rapidly across that tiny synaptic cleft and seek out specific receptor proteins on the membrane of the next cell, which we call the postsynaptic element. The fit is highly specific, like a bespoke key in a lock. When the neurotransmitter binds, it causes a conformational change in the receptor protein, and that initiates a new electrical or chemical change in the receiving cell.
The signal has been passed, but we can't have the chemical signal just lingering in the cleft where the receiving cell would just keep firing constantly. How is the message turned off?
Termination has to be fast and precise to maintain the temporal accuracy of the signal. There are two main mechanisms for elimination. First, enzymatic degradation. Specific enzymes exist right in the synaptic cleft to rapidly break down the neurotransmitter. The classic example is the enzyme acetylcholinesterase, which swiftly cleaves the neurotransmitter acetylcholine.
And the second method.
Reuptake. Specialized transport proteins embedded in the presynaptic neuron's membrane, or sometimes in nearby glial cells, actively pump the neurotransmitter molecules back out of the cleft and inside the releasing cell for recycling and repackaging into new vesicles. Both methods ensure the signal is brief, precise, and ready for the next impulse.
This brings us to a crucial concept in neurophysiology, one that often trips people up when they first learn this material. The effect of the synapse is entirely dependent on the receptor, not the neurotransmitter itself. The same chemical can excite or inhibit the target cell.
This is the rule that unlocks complex signaling. The effect whether the target cell experiences an EPSP or an IPSP is defined by what the receptor does when the ligand binds. To understand this, we categorize the receptors into two functional classes based on their speed and mechanism.
Let's start with the fast, direct communication, ionotropic receptors.
An ionotropic receptor is functionally identical to the ligand-gated ion channel we discussed earlier. Its mechanism is direct activation. When the neurotransmitter binds, the receptor protein immediately changes shape, and the channel pore opens.
So if that channel lets in a positive ion like sodium, the result is an immediate, fast EPSP. If it lets in a negative ion like chloride, the result is an immediate IPSP.
Exactly. The response is instantaneous and transient, measured in milliseconds. This is ideal for rapid actions like reflexes or fast motor commands. The key example is the nicotinic receptor for acetylcholine. When acetylcholine binds, the receptor opens, allowing NAO to rush in, causing an immediate depolarization.
So if that's the quick reflex pathway, what about the pathway that controls long-term changes like learning and memory?
That is the metabotropic receptor pathway. This involves indirect activation and a complex, amplifying internal cascade that leads to slower but far more prolonged and varied effects. The process is often called G-protein signaling.
Let's break down that cascade inside the postsynaptic cell.
The cascade begins when the neurotransmitter, the first messenger, binds to the metabotropic receptor protein on the cell surface. This binding triggers the receptor to activate an internal partner, the G protein. The G protein, which is essentially a molecular switch, then physically moves to and activates an effector protein, which is typically an enzyme inside the cell membrane.
So we have the external signal moving internal switches. What does that final enzyme do?
The effector protein catalyzes the creation of a new molecule, which is called the second messenger. Common examples include cyclic AMP or KMP or inositol triphosphate, IPs. The second messenger is where the signal truly amplifies and diversifies.
Why is this cascade first messenger to G protein to effector to second messenger? Why is that so much more important for long-term function than the simple ionotropic opening?
Because the second messenger can initiate a metabolic avalanche. While an ionotropic receptor only opens one gate, a second messenger molecule can diffuse throughout the cytoplasm and 1. Open or close dozens of distant ion channels, 2. Activate many different enzymes within the cell, or 3. Crucially, travel to the nucleus and change gene transcription.
Changing gene transcription means changing which proteins the cell is building, which is the definition of a long-term change.
Precisely. If a specific synaptic connection is repeatedly used, the metabotropic response might lead to the neuron building more receptor proteins or strengthening the physical connection. This long-term modification of the cell's physiology is a molecular basis for strengthening or weakening synaptic connections, which we hypothesize is the very mechanism of learning and memory. You need the slow, prolonged effects of the metabotropic system to achieve lasting change.
This beautifully illustrates why the receptor determines the effect. We classify neurotransmitter systems by their chemical composition. We have the cholinergic system, using acetylcholine. This is a perfect example because LE can bind to the fast ionotropic nicotinic receptor, causing excitation at the muscle.
Or it can bind to the slow metabotropic muscarinic receptor, causing diverse effects in the CNS and the autonomic system. Same chemical, two totally different outcomes.
Right. Then we have the simple amino acid neurotransmitters.
Yes, glutamate is the primary excitatory amino acid in the CNS because its receptors open channels that cause depolarization. Conversely, GABA and glycine are inhibitory because their receptors typically open chloride channels, causing hyperpolarization, actively suppressing neural activity.
And finally, the complex ones, the biogenic amines.
Amines, such as serotonin, dopamine, norepinephrine, they primarily act through diverse metabotropic receptor subtypes. Dopamine, for instance, has D1 receptors that are excitatory and D2 receptors that are inhibitory. When the same dopamine molecule hits a D1 receptor, it excites the cell. When it hits a D2 receptor, it inhibits it. This demonstrates the receptor determines the effect rule most clearly.
We have completed the entire journey of the electrical language, from the genesis of a small graded potential through the summation calculus, the all-or-nothing propagation down the axon, and the complex chemical handoff at the synapse. Now let's wrap this up by tracing the entire path of information flow from the outside world to a physical response, using that example of feeling hot water and deciding to move.
This is where we can really appreciate the scale of the system's speed. The entire sequence, from sensation to response, has to occur in a fraction of a second.
We start with the sensory input. The hot water is the stimulus.
Right. Specialized thermoreceptors in the skin detect the change. The heat causes a physical distortion or change, opening mechanically gated channels and initiating a graded potential. If the water is hot enough, that graded potential is large enough to drive the sensory neuron to the -5 mV threshold at the initial segment.
The action potential fires and enters the CNS.
The action potential propagates quickly along the myelinated sensory neuron axon, entering the gray matter of the spinal cord. It releases a neurotransmitter, triggering a new electrical event in the next neuron. This signal then ascends the sensory pathways, passes through the thalamus, the brain's grand central station for sensory information, and finally reaches the cerebral cortex for conscious awareness. Sensation complete.
Now for the critical thinking phase, integration.
Once in the cortex, the integration areas compare the information. Ouch, that's too hot. With your current goals, I need to finish this shower. And memories, I hate being scalded. This complex process culminates in the motor command, turn the cold water knob.
The brain sends the command back down.
The motor plan originates in the precentral gyrus, often called the motor cortex. The command is transmitted down the spinal cord by the upper motor neuron. This upper neuron synapses in the spinal cord onto the lower motor neuron.
The lower motor neuron is the final common pathway. It carries the physical command out to the muscle.
The axon of that lower motor neuron travels out into the periphery and terminates at the skeletal muscle fiber via a highly specialized junction, the neuromuscular junction, or NMJ. This is the ultimate non-negotiable chemical handoff.
Let's detail that last fast action step where the nerve becomes movement.
The motor neuron's action potential arrives at the terminal, opens the voltage-gated Ka channels, and releases a massive quantity of acetylcholine, or ASHWA, into the synaptic cleft. ASH binds specifically to the ionotropic nicotinic acetylcholine receptor on the muscle cell.
Unlike a typical neuron, the synapse doesn't mess around with summation.
Absolutely not. The amount of ASH released is so large and the density of receptors is so high that the resulting graded potential in the muscle cell, known as the end plate potential, is enormous. It is always powerful enough to immediately initiate a muscle action potential, which spreads along the muscle fiber membrane, leading directly to muscle contraction. Your hand successfully turns the faucet, and immediately that command is eliminated by acetylcholinesterase, ensuring the muscle can relax instantly when the firing stops.
It's an elegant, breathtakingly fast series of conversions. From mechanical energy, the heat, to electrical energy, the graded potential, to a digital spike, the AP, to chemical energy, the neurotransmitter, and back to electrical energy, the muscle action potential, all culminating in movement. The sheer complexity packed into that sub-second response is phenomenal.
The wonder, as you know, lies in the millisecond precision of these electrical and chemical conversions. We've demonstrated that all of our ability to sense, integrate, and act rests upon the structural integrity of the complex proteins, the Nyoko pumps, the voltage-gated channels, the receptors that manage ion flow.
So what does this foundation tell us about the future of neuroscience research, particularly concerning neurodegenerative diseases?
Well, the source material points toward a convergence in understanding diseases like Alzheimer's and Parkinson's, linking them increasingly to a shared underlying issue, proteopathy. This is the concept of proteins behaving badly, misfolding, aggregating, and accumulating in the nervous system.
Meaning the very structures we relied on to maintain that precise 11 millivite AVD charge and facilitate the synapse are beginning to fail physically.
Exactly. For example, Alzheimer's is characterized by the accumulation of toxic proteins like beta amyloid and tau. If you consider what we discussed in detail, that long-term memory and learning rely on the subtle, prolonged responses initiated by metabotropic receptors to strengthen synapses, requiring vast, complex metabolic changes, enzyme activation, and gene transcription. It creates a terrifying connection.
If the underlying machinery responsible for building and maintaining the proteins necessary for metabotropic signaling, the proteins that strengthen the connections that form our memories, begins to clog up and fail due to misfolding, the system loses its ability to adapt.
You lose the ability not just to communicate quickly, but to fundamentally change and remember. The precision required for the instantaneous action potential is maintained by thousands of complex channel proteins and pumps, all of which are built and maintained by these metabolic networks.
Right.
Proteopathy represents a physical, structural challenge to the very language of the nervous system. The electrical signal might still fire, but if the synapse cannot be maintained, strengthened, or appropriately terminated, the system degrades into silence and chaos.
The ultimate fate of our cognitive self, our memories, and our ability to integrate complex information may ultimately hinge on whether those specialized channel and receptor proteins can simply hold their complex three-dimensional shape over a lifetime. That is a truly mind-boggling frontier.
It is the intersection of biology, physics, and consciousness. We must continue to unravel that critical connection between the electrical life of the neuron and the structural integrity of the proteins that make it all possible.
This has been an incredibly comprehensive and deep dive into the electrical language of the nervous system. Thank you for guiding us through this foundational material and for showing us not just what happens, but why it matters.
My pleasure. It is a system worthy of every moment of study.
To you, our listener, thank you for joining us on the deep dive. We hope you walk away feeling thoroughly informed about the amazing electrical network governing your every thought and action. Until next time, keep diving deep into the knowledge you seek.